Today we shall discuss about the structure and function of plasma membrane. It is very important for those students who are preparing for NEET medical entrance and 10+2 students as well. First, let's have a look at its introduction. A plasma membrane encloses every type of cell, both prokaryotic and eukaryotic. It physically separates the cytoplasm from the surrounding cellular environment. Plasma membrane is a living, ultra-thin, elastic, dynamic and selective transport barrier. It is fluid mosaic assembly of molecules of lipids, that is phospholipids and cholesterol, proteins and carbohydrates. Plasma membrane controls the entry of nutrients and exit of waste products and generates differences in ion concentration between the interior and exterior of the cell. It acts as a sensor of external signals and allows the cell to react or change in response to environmental signals. Let's have a look at the ultrastructure of the plasma membrane. Under electron microscope, plasma membrane appears as a trilaminar membrane consisting of number 1, an outer dense layer of about 2 nm formed of proteins and polar ends of phospholipids. Number 2, an inner dense layer of about 2 nm formed of protein and polar ends of the phospholipids. Number 3, a middle layer of sandwiched between the two dense layers of about 3.5 nm formed of non-polar fatty acid chains of phospholipid molecules. Number 3, its chemical composition. Have a look at the chemical composition of plasma membrane. The plasma membrane is composed of globular proteins, lipids and a small percentage of carbohydrates, that is oligosaccharides. Their proportion in the plasma membrane of different types of cells varies greatly. Thus, chemically, plasma membrane and the other membranes of different organelles contain proteins, lipids and carbohydrates, but in different ratios. For example, in the plasma membrane of human red blood cells, protein represent 42%, lipids 40% and carbohydrates 8%. Have a look at the lipid of plasma membrane. The main lipid of plasma membrane are phospholipids, cholesterol and galactolipids. Phospholipids are asymmetrical, elongated molecules, amphipathic or dipole in nature. Each molecule consists of number 1, hydrophilic water-loving or the polar end, and the hydrophobic water-hating or the non-polar end or lipophilic end. The hydrophilic or the polar end is the rounded head and is usually formed of choline phosphate. But the hydrophobic or the non-polar end, also called lipophilic end, comprises of two molecules of fatty acids that are chemically bound through their carboxyl group to the glycerol backbone. Glycerol in turn is connected with the phosphate of head end. These two fatty acid molecules are called hydrophobic cells. In the plasma membrane, the non-polar hydrophobic fatty chains of the phospholipids meet and develop in the middle line, while the polar hydrophilic heads are directed toward outside the membrane. Types of phospholipids found in plasma membrane. The phospholipids in the plasma membrane are of two types. First, neutral phospholipids and second, acidophilic lipids. Have a look at the neutral phospholipids. Neutral phospholipids form the major portion of membrane phospholipids. Their polar heads are without any electrical charge at neutral pH and are tightly packed in the lipid bilayer along the cholesterol. The most common neutral phospholipids of plasma membrane are phosphatidyl choline, phosphatidyl ethanolamine, lecithin, cerebroside, and asparagine. Acidophilic phospholipids have polar heads having negative charge and are associated with the proteins by way of lipid-protein interactions. These constitute 5 to 20% of phospholipids. For example, phosphatidyl ionositol, phosphatidylserine, phosphatidyl glycerol, thiazolipin, and sulfolipids. The distribution of phospholipids in lipid bilayer plasma membrane is partially asymmetrical. Have a look at the nature of the proteins found in plasma membranes. Plasma membrane contain three different classes of proteins: structural proteins, enzymes, and carrier proteins. Structural proteins form the backbone of the cell membrane. They have little catalytic activity and are extremely lipophilic. The plasma membrane consists largely of structural proteins. Enzymes form the major component of many membranes and are catalytic proteins. The structure of enzyme varies from membrane to membrane. Carrier proteins transport molecules across cell membrane without being permanently altered in the process and against concentration gradient. The plasma membrane proteins fall in two main categories: intrinsic or integral proteins and extrinsic or peripheral proteins. The former are firmly associated with the membrane, while the latter have a weaker association and are bound by electrostatic interactions. In different types of extrinsic and intrinsic proteins and the structural components of plasma membrane and models of the plasma membrane will be discussed in the later phase. Till then, take care.